after each presentation, or you can just type your name, and then we will give you the floor and you can ask your question yourself. So, and also please just use the chat box for this purpose. Otherwise, it will be very difficult for us to find the questions. Classic housekeeping rules nowadays, when everybody moved to electronic meetings, is that when you're not speaking, turn off your microphone. When you speak, turn on your microphone. And you can also use the camera when speaking. We have the presentations, or some of the presentations here, so if there are problems, I saw Sylvain that it was a bit difficult with yours, so maybe we share it from here. Let's see how it works out. But I leave now the floor to Ines Steffens.
Yeah, good afternoon, everyone. It's a pleasure to be here. I'm Ines Steffens. I'm the editor of Eurosurveillance, and in my job at an international public health agency, I've had the privilege over many years to meet very inspiring and bright people working in the field of public health and communicable diseases, and specifically. And among these, I met nice colleagues, and some of them became closer colleagues and friends, and Marc is obviously one of them, because we met and we asked all the speakers to let us know when they met Marc for the first time. And when I thought about when I have met him for the first time, actually it was at a party at a friend's house when Marc had just come to Stockholm with his family, and we realized the two kids that were running around and being a bit bored were our daughters. So we found some commonalities here, and over time we found more commonalities. We found professional commonalities. We were both members of the Ecology Scientific Committee for many years, and we shared the same views and vision on public health and the direction that we thought we would need to go. And also we had both the same feeling and strong feelings about how to scrutinize scientific evidence. And so for one short period of time, we both were members in the same unit, and this was then the unit headed by Andrea Ammon, who's then the deputy to the director and head of unit for response management, for resource management and coordination. And we all know Andrea Ammon is today the director of ECDC. Without further ado, I would like to pass on to Andrea, our director, to give us more insight into and his influence, chief microbiologist, ECDC. Andrea?
Yeah, thank you very much, Ines and Daniel, first of all, for organizing this together. Thanks also to our guest speakers. I know they're all busy men, and I find it very touching that you agreed to speak this afternoon. Dear Marc, this is to say a proper farewell to you, at least for the time at ECDC. This retirement comes after a very long and shining and successful career, and I'm very happy that you have chosen ECDC for the last 10 years to be the chief microbiologist. I think you started in almost 10 years ago at on 1st of October 2010. And this was the time when ECDC, together with the national microbiological focal points and the disease networks, really embarked under your leadership on the journey of molecular surveillance, in particular, then also leading to our strategy on whole genome sequencing and the gradual integration of sequencing into surveillance. So not only for outbreak situations, but really to the routine surveillance. I think this was a very important part of your contribution to ECDC. I still believe that even more far-reaching was your work together with our national microbiological focal points on the EU Lab Cup system, because this is a system that allows the member states to capture the status of the laboratory services, the capabilities and capacities in the countries, but also to monitor this. It is, when you look at it, a very complicated system. And I am sure if the colleagues in the member states that actually have to fill it in hadn't worked so much together with you, I think we would have never got this off the ground. But so we had really many iterations and a constant improvement over the years. And it is not only really applied here in the EU. We have now adapted it for the enlargement countries, and I know we had requests also from outside of Europe, from Canada, from African countries. So I think for me this is really a very an outstanding piece of public health value. You were also, of course, involved in our representative, our interlocutor with the DG RTD projects, and in particular Prepare, now Ecology and Compare. But also you put together this observership of ESCMID and ESCV young members for every year that they come to ECDC, and I think this has been a real success story. They are really bright colleagues that have a good interaction with us, and I always enjoy meeting them. So this whole journey that we embarked is now, of course, meeting COVID and SARS-CoV-2. And I think this pandemic has highlighted and accelerated some elements of the transformation that we are experiencing right now in our work, both on the epidemiology and the microbiology side. I believe that in 5 to 10 years, the ways how we collect data, how we analyze them, interpret them, report them, communicate them will be completely different than what it is now. And you know, when I said the pandemic has highlighted developments, we were always talking about in the past, well, since I came here, since I became director since the last 3 years, about how we can harness the advantages of e-health. And here, what we have seen is that, in particular, in the first phases of COVID, the way how we have organized surveillance is just having too many steps. The necessity of human input that was not there, because the people that do this input had other things to do. They had to find cases, trace contacts, monitor contacts, and the surveillance was sort of the thing that came after this. So here it's very clear that the digital solutions could help us to be more timely, to have the data more timely. The other thing that where I think it has really accelerated the developments is with the apps. I believe what we have seen in this 8.5 months in development and field testing of the Corona apps is something that we would have never ever been able to put in place in such a short period of time. The fact that it may not have worked in the first attempt is not discouraging to me, because we learn from this and the next generation will be better. We see that the in microbiology, the sequencing has now really, I mean, it's taken over. And when I remember that I had the first group discussing sequencing, molecular typing as part of surveillance in 2007 here. And when we went to the advisory forum, they nearly threw me out because it was met with a lot of hesitation and resistance. But now I think it is accepted. The roadmap is consecutively implemented, and I think you helped a lot to actually promote this. We have now, how should I say, also recognized, I think not only we, but also colleagues in the member states, that there are distinct advantages to sequencing because it allows a better detection of clusters and a characterization of clusters, a clearer definition of outbreaks, and also here the link to the pandemic, the possibility of detecting reinfections with slightly variant strains is also only possible due to the whole genome sequencing. Last thing that the pandemic, and that links a bit to the other topics that are mentioned or will be mentioned by our speakers in the following. The pandemic has highlighted the vulnerability of our health system. The focus of the health system on COVID has inevitably led to a negligence or less focus on other diseases. And the one of the diseases, I mean, is infectious diseases. The big priorities that we had on vaccine preventable diseases and antimicrobial resistance, we don't know yet the fallout of the fact that they were not as given as much attention as they should have been. We have seen that there's a lot to do in terms of preparing our hospitals, in terms of search capacity for staffing, in terms of stockpiling of material and equipment. But it has also highlighted that health in all policy is not just a concept on paper, but it is, I mean, it's reality. And I think that has been driven home now by the pandemic in a much more forceful way, that I hope that also in the following time it will not be forgotten. So I think, Marc, the work that you did in the last 10 years, but also before, and what is happening right now, there is a link and there is a path that you have been going, and I think you can be very proud of what you have achieved in your life. So I hope you enjoy the next 2 hours, and I will have an opportunity to give some more personal remarks at the end. Thank you for now, and I hand over to our moderators.
Thank you, Andrea. We move to the first speaker of today, and that is Sylvain Brice from Institut Pasteur. Sylvain is interested to cover microbial population biology and evolution of pathogenic microbial species and its application to diagnostics, epidemiology, and public health, so very close to Marc's interest. Sylvain has also been involved in developing strain nomenclatures for various pathogens, and he met Marc during an FP5 funded project some 20 years ago. So they have been colleagues ever since. So Sylvain, I give you now the floor. So let's now see if your connection works better. Otherwise, we run the slides from here. Sylvain, please.
Hi, do you hear me? Yes.
Yes, loud and clear. We hear you, Sylvain.
Wonderful. So let me share So should I? We can't display your shared content. Make sure you have allowed permission to share. Yeah, I have to find the right.
Sylvain, I'll try from here. Let me know if you see it.
Yes, it's open on my desk, so I should be able to share it.
I think your connection might be a bit, because the picture is quite, it's not very clear. So we might have
you see it, but you. Well, it's there is a small circle going around. So it's doing something.
You see the slides now? Can you?
Otherwise, yes, please share the slides. I could see them, and I'll just ask you to.
We will share the slides from here, and Daniel is just doing it, okay?
Yeah,
As
fine.
soon as you see, you start.
All right. Yes, I see them. So thank you very much, and it's really a great pleasure, Marc, to be here and to celebrate this very important moment together with you. I also see there are many colleagues connected, and I say hello to everyone. Many friends of many years. Marc, you really created a community of great colleagues and friends. And you asked me to talk about a very challenging subject, which is written on this slide, and so how ecology, vaccines, and antibiotics are driving the evolution of bacterial pathogens. This is obviously a very challenging title, and I have tried to think how I could make a 20 minutes presentation on such a broad topic. So in the next slide, you'll see I have kind of simplified the question into how the different microbial ecologies, let's say, impact the effect of human interventions on the evolution of pathogens. And when we talk about human interventions, I will obviously focus especially on vaccination and how vaccination does impact the evolution of pathogens, and also a little bit on antimicrobial therapy. So next slide, please. I guess we all know that evolution is a mix or combination of diversity-generating events like mutation and recombination, together with neutral genetic drift or selection, which will impact the frequency of different alleles or genotypes in the population. And obviously, there are other processes such as the demography or migration, which are important in the evolution of pathogens. So in terms of public health impact, and this is really what we are interested in, I guess, the evolution we are looking at is really the emergence and spread of types, and especially antimicrobial resistance and vaccine escape in terms of and clinical. So next slide, please. So I thought it would be useful to kind of divide ecology, the broad study of ecology or ecological science, into different subtopics or questions. And I've listed 4 questions here, a bit schematically, obviously. So the first question is, who are we talking about when we study the ecology of a species? It's important to define clearly the species and have a clear taxonomic framework, but also to look at the population structure of these taxa and to be able to define strains. And Marc, you have been quite instrumental in this area, as we all know. The second question that is included in ecology in general is where. So where are the strains or the species distributed? Where do they find them? Where do we find them? What are their reservoirs? And how do they transmit? The next question is why. And this has to see with adaptation of the organisms, and the molecular mechanisms of adaptation, for example, to a specific environment, a specific niche. And finally, we can also ask the question, what? Meaning, what do they do? What is this microbe doing in this niche? So this can be interactions with the microbiota or host benefits, for example, on the immune system development in humans or the host pathogen relationships, especially in the context of disease. So in the next slides, obviously, I will not treat all these questions. I will focus on what is generally considered when we talk about the ecology of pathogens, which is the distribution. So the second question, the where question. Where are the species found, and how does this affect actually the human interventions? Next slide, please. So I guess the microbial lifestyle impacts the way pathogen emerge in very important ways. If you consider a specialized pathogen on the right side of the slide, where this pathogen, for example, is only found in humans, either in carriage or in disease, and if you exert a selective pressure, for example, by vaccination or by antimicrobial therapy, on the population of pathogen in this human population, then every genetic change that will be selected, for example, a resistance mechanism will be selected, but also it will be passed on to the next generation because the transmission is within the population, within this particular niche. And so you have a kind of vicious circle where you will have an accumulation of selected genotypes. And this is really what we call directional selection. On the other hand, if you are a generalist pathogen with many different reservoirs, like environment or animals or food, and you exert the selective pressure only on the human colonizing or infecting population, then the mutations that are selected by these pressures will not necessarily be successful because they won't be transmitted to the next generation, especially if infection or colonization in humans is always driven by exposure to environmental or food sources, for example. And so in this case, we don't expect any impact of antimicrobial treatment or vaccination. And I have kind of organized on a scale from generalist to specialized 4 different pathogens, which I've been studying in the past, and some of which I'm still studying. So on the left, we have Listeria, which is a generalist pathogen. We find it almost everywhere in the environment and obviously in food, which it contaminates. And on the other end of the spectrum, I have placed Volatile Hypothesis because it's a highly specialized human pathogen. And you can see Klebsiella pneumoniae and Corynebacterium diphtheriae somewhat in the middle, because, for example, diphtheria is mainly a human pathogen, but we do find it in some animals occasionally. And Klebsiella pneumoniae obviously is a very generalist pathogen, but also circulates among humans, unlike Listeria. So a single scale is probably not the best way to represent the complexity of lifestyles, but this is just to help the discussion. Next slide, please. So the first example I will consider is Volatile Hypothesis, and because this is a human-only pathogen, it's a more simple example to consider first. Next slide, please. So the way Volatile is controlled is mainly through vaccination. Until the 90s, we were vaccinating using whole cell vaccines, and this has switched to acellular vaccines between 90 and 2000 in different northern countries of the Western world. And acellular vaccines are basically composed of 1 to 5 antigens, which you can see listed here. So the primary aim of the vaccination is really to control severe cases in infants. And it's important to know that vaccination, at least with the acellular vaccine, does not eliminate colonization. And also, neither the infection nor the vaccination will protect you for more than 10 or 15 years against pertussis. The other control we have on pertussis is azithromycin or other macrolides, which are used in case of infection, but also to prevent colonization and transmission prophylactically. Next slide. So the epidemiology of pertussis has shown in many countries that pertussis is resurging. Of course, it's resurging after a sharp decline. I'm not sure you can see my the pointer, but If you see on the left side, you have the situation in the US from 22 to 2012, 1922 to 2012. And you can see that initially the vaccination was extremely effective in controlling the disease. But we do observe a resurgence in the US and many other countries, such as Australia or the Netherlands, since a few years, approximately the 1980s. So the question is why, and there are many different factors, including strain evolution, as has been shown. And the next slide illustrates that very well. So this is a cartoon from the classical study of Barth et al., coordinated by Fritz Moy and Julian Parkhill. And this has really shown that the evolution of volatile hypothesis has happened in part through a succession of mutations, specifically in the genes coding for the acellular antigens. And this is the case for pertussis toxin, for the toxin promoter itself, and for Fimbriae type 3, as well as pertactin. And what we observed here is that the mutations that happened during the whole cell vaccination era, mostly, but also led to a divergence from the vaccine strain. The vaccine strain is marked with a T in the left figure. And so you can see the temporal evolution of the frequencies of the different alleles for two genes that are illustrated here. And this is a rapid selective sweep that has been observed, for example, for PTXA1 allele. That is the first mechanism of evolution of pertussis through antigenic divergence. And the next slide. The next slide is more focusing on one of the antigens, because a second mechanism of evolution to escape vaccination in pertussis is through the loss altogether of the production of the antigen. And this is mostly observed for pertactin. The red line illustrates the proportion of pertactin non-producing strains over the years. And you can see a very rapid increase since the use of acellular vaccination that started in 25 in France. This is French data. And we occasionally observe the loss of other antigens like PT, the pertussis toxin, or Fimbriae, but this is really anecdotal compared to pertactin. And the loss of pertactin has been observed in multiple branches of the phylogenetic tree, indicating a very strong selective pressure to lose the expression of this antigen. Next slide. Another illustration of the importance of pertactin loss on perhaps the fitness of the strains in a population that is vaccinated against pertussis is this cartoon, which shows that the longer a country has been vaccinating against pertussis, the higher the rate of pertactin negative strains is observed. This is data from 2012 to 2015. You can see France had a prevalence of 25% of pertactin negative strains, and today we have a prevalence above 50%. So this has still continued to increase. So this is completely consistent with the continuous selective pressure in countries that use pertactin as one of the acellular vaccine components. I'm hearing some voices. Maybe someone should shut the microphone off. Next slide, please. So regarding experimental data to demonstrate the effect of a selective advantage effect of the loss of pertactin, there has been experiments in mice performed in the lab of Nicole Guizot in Pasteur, as well as vaccine efficacy data in clinical studies, which have shown that the loss of pertactin should bring directly a selective advantage in vaccinated individuals. There is also evidence from in vitro and clinical studies that the fitness cost of the loss of pertactin is very limited or non-existent. This has been shown in macrophage cytotoxicity assays, and also by looking at the clinical severity of strains with or without the pertactin production. Next slide, please.
Can I ask again that you check that you have your microphones muted if you're not speaking? Sorry, Sylvain. please continue.
Yes, yes, there is a background noise now. Next slide, please. Okay, I will skip this slide because I'm a bit short on time. So concluding on pertussis is that there is a very clear impact of vaccination on the population evolution. But obviously, this raises a lot of questions on vaccine efficacy. It can be expected that the efficacy is diminished against strains that have lost protectin, basically. And there are outstanding questions that remain. For example, the spread of strains across countries could blur a little bit the effect of local selection. And so this is also a question we are investigating at the moment. And clearly, this all highlights the importance of continuing the surveillance of pertussis. Next slide, please. So I would like now to turn to a second example, which is quite more complex, which is Klebsiella. So Klebsiella clearly is a ubiquitous pathogen. We find it everywhere. Some data show up to 5 to 10% of prevalence rate of Klebsiella in soil, for example, or in plants. And in animals, it can go up to more than 50% in gut carriage. That's also the case in humans. And what is interesting is that most infections by Klebsiella in humans and in animals, to some extent, are opportunistic. So they happen in patients with risk factors, which are well known. Next slide, please. So the issue of antimicrobial resistance in Klebsiella is really, I would say, dominated by two main themes, which really are dependent on the ecology of Klebsiella. So the first body of evidence is that there is a high rate of antimicrobial resistance in the hospital. And this, we know, is due to the circulation of hospital-adapted strains, which are subjected to the selective pressure by antibiotics in the hospitals. This has been shown by countless number of studies. And one example that is quite archetypical is ST258, which carries black APC, which has emerged in the mid-90s and now has spread all over the world almost. So the question for these kind of clones, obviously, is will they disseminate into the community? And so far, we see a little bit of carriage in people who are discharged from hospitals. But apparently, ST258 really doesn't colonize a large proportion of people, nor animals or the environment, because it has never been found in the environment. So we are dealing here with a complete effect of a hospital niche that is quite special and perhaps quite disconnected from the overall ecology of Klebsiella. And then on the other hand, Klebsiella pneumonia is exposed by its generalist lifestyle to many different kinds of bacteria. So a huge diversity of strains can exchange genes with Klebsiella, basically. And so this provides Klebsiella the opportunity to sample genes and pick them up. And because it circulates between the environment and humans, it can also bring these genes into the human clinical settings. And so this is, for example, exemplified by MCR1, which has been early detected in Klebsiella almost at the same time as E. coli, and is quite prevalent, or more prevalent than in E. coli in many settings. In other words, Klebsiella samples the diversity of genes in the environment and can sometimes, although this is rare, bring them into the clinic and become a public health issue. Next slide, please. So this has been kind of very well advocated by the group of Kat Holt in Australia, where they have shown that a number of genes that are quite relevant for clinical antimicrobial resistance have been first described in Klebsiella. You can see the table here: SHV1, SHV2, QNRs, KPC, OXA48, etc. And so they have used the allegory of the canary in the gold mine, which means that Klebsiella can be used as a kind of sentinel organism to detect early novel genes that might be important for clinical resistance. On the other hand, Klebsiella is able, due to its broad lifestyle, to transmit these genes to other pathogens, including Enterobacteriaceae or non-fermenters, for example. So this is a very significant role, an ecological role, if you wish, of Klebsiella that is important to underline, I guess. Next slide, please. So the study of Klebsiella ecology has been quite difficult due to the broad distribution, but also due to a number of shortcomings, including difficulties in the classification of strains. So the taxonomy was ambiguous until recently, and also a lack of methodological approaches that were harmonized but also quite efficient. And in the recent years, there has been quite good progress on the different aspects of Klebsiella ecology studies. So there have been taxonomic updates, there have been selective media developed, qPCR to detect Klebsiella in complex environments, MALDI-TOF to identify Klebsiella precisely, but also bioinformatics tools, for example, to characterize the strains. And in the recent years also, there have been a number of networks funded to study the Klebsiella ecology on a broad scale. They are listed here, and these have meant that are now large consortia that are using Klebsiella as a model to study its ecology, and we can expect large data sets to be produced soon. Next slide, please. So one important theme in Klebsiella ecology and population structure is really that the population structure is highly structured into a number of sublineage that we call clones, which are quite distinct from each other and quite homogeneous within each other, within themselves. And so typically we distinguish hypervirulent clones, which are observed in general in community-acquired severe infections. And they are the blue clones in this slide. And then on the other hand, you have multi-drug resistant clones, which typically happen in hospitals. So this has been a theme, a dominant theme until recently, and now we are observing the emergence of clones that combine these two characteristics, the so-called dual risk clones or high risk clones. And this is now becoming a serious public health threat. So one important question to perhaps mitigate this evolution is to understand better the ecology of high-risk clones. Next slide, please. So until now, the ecology of high-risk clones is not so well understood. In this slide, I'm presenting the metabolic diversity of Klebsiella. So we have looked at a diverse set of strains, including hypervirulent and multi-drug resistant strains. And the take-home message is that there is a huge metabolic capacity of Klebsiella. It can really use a high number of substrates, but also there are interesting differences between the different clones, and this really warrants future studies on the ecology of the specific clones. Next slide, please. So one of the large studies on the ecological niches of Klebsiella has been carried out together with Bicham Heun in Pasteur, and we have published that recently. We have looked at human gut carriage in pregnant women because there is a high relevance to neonatal sepsis, but also in 3 low and middle income countries. And basically what we have found is that the prevalence of Klebsiella is quite high in gut carriage. This is completely asymptomatic gut carriage, and it reaches 60% in many settings. So that's an interesting observation, because until now it was believed to be between 5 and 25% of carriage. So clearly we have been using probably a more sensitive method. The second big result of this study was that there is a huge diversity, and almost every ST was unique. Every strain was unique, and we only found 2 women, although these women were sampled were living in the same area, in the same neighborhoods. Only 1 pair of women out of 900 or out of 500 that were positive for Klebsiella, carried the same strain based on a genomic definition of strains. So clearly there is I call this a sampling dilution effect. And it's hard to understand the transmission and ecology of specific clones if you never sample them more than once or twice. And so this is a huge challenge for Klebsiella ecological studies, at least at the level of the clonal groups. Next slide, please. I'm almost finished. So coming back to the initial slide where I was listing 4 questions encompassed by ecology, I would like to just underline the first question, which is who, and the taxonomic definition of strains. Next slide, please. Just wanted to mention that we host at Pasteur a repository of the genomic and genetic diversity of Klebsiella strains. This encompasses not only Klebsiella pneumoniae, but the related species, which you can see here. And this resource has been extremely useful to create a unified language on the genotypes of Klebsiella, which then enables people to work together on the specific ecology or biological questions around specifically interesting clones. And so in only 5 years, the number of STs in this database has more than doubled, which illustrates the rapid speed of expansion of our knowledge in Klebsiella ecology at the moment. Next slide, please. All right, this brings us to the most important part of this presentation, which is about Marc himself. Marc has been quite instrumental in the implementation of so-called library typing system, as he called them quite early, actually. This is back in '98, as you can see from this publication. And Marc has been a strong advocate of standardized and reproducible typing methods so that we could do molecular epidemiology not only at the local scale, but also at the global scale for understanding the population structure of pathogen and long-term evolution. And Marc has always been advocating as well, I think this is very important, to work together between the public health sector and the more fundamental studies in evolution or ecology of bacterial pathogens. And this is clearly illustrated in this sentence, which comes from one of his papers on these topics. Next slide, please. So another very important contribution of Marc has been the creation of IMBEM meetings. They were called IMBEM, I think, at the beginning, because this was only bacterial, but then they realized viruses and parasites were also interesting to study. I'm not sure why. But anyway, Marc has, I guess, organized the first IMBEM meeting in Brussels in '87. So this is more than 30 years ago. And since then, these meetings have been always organized every 2 or 3 years and are quite successful. So this is really a very important contribution by Marc, not only for science dissemination, but also by creating a community of good colleagues and friends. And many of you are around this, this symposium today. That's great to see. Next slide, please. And this will be, I guess, my last slide. So this is, you know, in the old times, we used to meet physically. So this here you can see, you know, Marc Struelens during a physical meeting. So this is what I call a physical meeting experience. And you can see the pleasure that was provided by such meetings to Marc. And yeah, these were times where we used to meet in person, you know, to discuss things, and we could touch each other, we could talk to each other in the face, we could even cough to each other, and that was these were great times, which obviously we have a bit forgotten in this moment. But I think Marc really it was enjoyable to meet you in person and to you know, for you to create all this community. I think this has been a great achievement, and on behalf of all these communities that are listed here, I would like to give you a big thank you, and hope to meet you physically again at some point. Thank you very much for the attention.
Thank you very much, Sylvain, for this very, very nice presentation. Can I again remind you that you have the chance to ask questions in the chat box, and we will try to bring them up or you ask for the floor, and you can ask them not in person, but in by electronic means. So first, I ask Mark, do you have any questions to this presentation? Over.
Can you hear me? Can you hear me?
Yes, we can, Mark.
Thank you, Daniel, for opening the floor. Thanks a million, Sylvain, for this fantastic talk and your very warm words at the end. Indeed, we missed close face-to-face chance to talk, but let's do it through this channel. I had one question for you. To what degree, considering the distribution of human pathogen or polycystic pathogens from the kind of specialist parasite life to the more kind of generalist commands of a opportunistic pathogen, what is in your view, the relative strength of horizontal gene transfer in shaping the evolution, especially currently under human pressures such as chemical and antibiotic pollution of both the human microbiota, but also the worldwide microbiomes. Over.
Thank you, Mark. This is a great question. I would say horizontal gene transfer occurs irrespective whether you are a human specialized pathogen or a generalist pathogen. I think in both situations you can find clear examples of horizontal transfer. Let's take the example of the pneumococcus, for example, where this is also a specialized pathogen observed only in humans, and the issue of horizontal gene transfer is quite clear, especially in the face of vaccination and escape of the strains from the main antigens that are included in the vaccines. So on the other hand of the spectrum, Listeria, for example, which is mainly an environmental pathogen, doesn't recombine much. We know it's quite clonal. So you can find the counter examples in each of the settings. I would say the only exception perhaps is the obligate pathogens, which are intracellular. It's well known that obviously for obvious reasons of probability of encounter with mating types, say with quotes, they don't recombine much, and they evolve by loss of genes mostly. But otherwise, I would say horizontal gene transfer is quite important, both for antigenic switch in the face of vaccination or for the acquisition of antimicrobial resistance, which I guess is horizontal gene transfer is the main mechanism for that.
Thank you, Sylvain. We learn as we go here, and we see that maybe if you type a question in the chat, that you also provide your full name, so we can also say who asked the question. But there is one here for you, Sylvain. Why do you think the MCR gene is more common in E. coli than in Klebsiella?
I don't think that. I just read it, so there is evidence in the literature, at least in some settings, but I don't have the figures with me right now. I think it was I wouldn't remember the reference where I read that. Some people around this table might know more about this. Obviously, I'm not in a clinical setting. So.
Thank you, Sylvain. And one last question.
Well, if I may, Daniel, on that point, I guess one of the questions is why would there be more prevalent MCR gene in the environment and food animals than in humans? Is that the question?
Yeah. So.
Yeah,
in that case,
to the agricultural use of colistin, especially in some region like China and Asia, where it's been massively used, and other parts of Europe is one thing. So there might be more selective pressure in the agricultural environmental reservoir and food animals than in human medicine. Over.
Yeah. And I could add to that Klebsiella is quite efficient at acquiring plasmids and maintaining them. It seems that the fitness cost of novel plasmids in Klebsiella is rather low compared to E. coli and other organisms. So it could also be part of the explanation that MCR1 and the other NCR genes, by the way, have diffused and disseminated in Klebsiella in many different plasmid backgrounds. So maybe that's part of the explanation as well.
Okay, one last question for you, Sylvain, and that is, you mentioned that hypervirulent Klebsiella was most often found in the community. What has really up to now stopped that one from acquire the full resistance pattern and also be a really successful pathogen in those other environments where there are a lot of antibiotics over?
Well, we don't really know the ecological niche, let's say, of hypervirulent, so-called hypervirulent clones. We know some of them are available are found in pigs, for example, ST25, and ST23, the typical hypervirulent clone from Taiwan and Asia in general, is quite frequently found in gut carriage in these populations in Asia. Now, why don't they acquire more antimicrobial resistance? I guess this is just a matter of time, because we do see these clones acquiring resistance, and we do see resistant clones, which are typically, and this has been shown by the group of Kat Holt, the resistant clones are more prone to acquire new genes and new plasmids, especially. And so they acquire the virulence plasmid and become themselves hypervirulent clones, resulting in multi-drug resistant clones being also hypervirulent. The reverse phenomenon indeed has been less observed, seeing a transfer of antimicrobial resistance into hypervirulent clones has been more rare, but we don't really know why. Probably it's a question of niche and lower exposure to antimicrobials.
Thank you very much, Sylvain. And again, thanks for a very nice talk. I leave now the floor to Ines to introduce the next speaker. Ines.
Yeah. I have the pleasure to introduce Dr. Alexander Friedrich, a true European who grew up in Germany with a German father, Greek mother. He studied in Portugal, married an Italian, with whom he lives together in the Netherlands, in Groningen, where he heads the Department of Medical Microbiology and Infection Control at the University Medical Center. And as we asked everyone where they met Marc, and Alex will have more on that, but he echoed in a way what Sylvain said. Of course, they met in a European network. And of course, as a very young microbiologist in the early 2000s, he was fascinated and very much inspired by Marc, who was then already one of the senior figures in European public health microbiology. And he was so open and one of the forerunners in applying new typing methods and working hard on a common nomenclature and also on harmonization of PGFE protocols, in particular for MFSA and MRSA. And without speaking longer, Alex, I think I would like to leave you the floor. Please take over. Alex?
No, unmuted. Do you hear me?
Yes, we hear you.
Excellent. So thank you very much. Thank you for the honor of being invited to talk today here. Marc, we all have no time, but that was the moment that I had to be. And I see from the list of colleagues, friends that are here, that many others, and I'm so glad to feel you here in the same meeting, although I cannot all see you. I was asked to talk about the clinical microbiology part and in controlling antimicrobial resistance and the use for in patient safety. Here's my disclosure. There's no conflict of interest. In a time where we all, whatever we are doing, at least in clinical and public health, microbiology are dealing for 90% with coronavirus 2. I will talk about the other continuous existing challenge for healthcare, on the healthcare-associated infections and multi-drug resistant microorganisms. I like usually to ask in the beginning this question. Imagine you are living in the first floor apartment of a house and it erupts a fire on the roof. What will people do? Close the door so the fire will not reach your apartment. Close the water supply that you will have water, and then the others that have fire don't need it anymore. They have already problems. Or go there with water and protect the others in order to protect them and yourself. If we look to Europe and the distribution on a national level, we see that there's some fire. And there's some fire in the roof. And what are we doing? We cannot close the doors, and we should not. And we should bring the water to where the fire is. And I ask all of you and myself, are we doing that? We would like to do, but are we doing that as Europeans? And I think that we see also at the current crisis that there is another disease in our system, which is the disease of the diversity in systems, and that they are still not integrated enough. It's a demographic challenge because Europe, together with China, more than other continents, will be one of the oldest continents over 20, 30 years. It will be a nice world, so much cumulative age at the same time on the planet. A lot of experience of life. Will this experience be able to be transmitted to other generations by having healthy people by doing that? Or will we all becoming older and sicker and more morbidity, and with more morbidity, more intervention-associated infections? And by combating that, and in the One Health problem we all know, the antibiotic resistance problem that has been nicely described by many people, especially from the ECDC, the impact on the European situation and on the people. What are the strategies? And I will be talking from the clinical part, and this is how I understood that. And we all, of course, hope that there will be new antibiotics. But do we really need new antibiotics, or do we need smart antibiotics that know which microorganisms to eliminate and which not? Combinations of nanotechnology with biochemistry. But that's not everything, because it's the curative aspect of it. But the prevention aspect of it looks into using what you have in a better and optimized way. The classical resistance prevention, also a preventive act by doing optimal treatment. And that is, of course, only when needed. The transmission prevention, I call it the hygiene 1.0. It's the classical infection control. Diagnostic stewardship to be more personalized, having diagnostic possibilities that are not black box high throughput, but personalized to the patients that I have, giving me information of the patient from genetics, using metagenomics on the moment of the infection and follow up. And then the last is the hygiene 2.0, which I would like to call network prevention. I will talk not about all, but of some of them. But many times I meet people and they tell me, but it's so easy. The reasons for spread of antimicrobial resistance is so easy. We all use too many antibiotics, doctors, veterinarians, and whoever does it. There is a lack of hand hygiene, and if we would just have good guidelines and not so much unobedient healthcare workers that are not washing their hands or disinfecting hands, then the problem would be solved. I'm afraid that that will not work. That we say, oh, because in the Netherlands there is no problem in MRSA or in other microorganisms, the others just need to do that, or vice versa. You can talk about. I will try to explain that. Why is that probably not true and not so easy? But the question is, what have we done the last years, and what is the future? And sometimes it's good to go back and to read about ideas of important people of many years ago. And there are not so many people that have an interview, a one-to-one interview in Lancet Infectious Disease. But Marc Struelens had, as when he became president of the ESCMID, and today I represent also the ESCMID as treasurer of the executive committee. Marc was president of the society in 2004. And he was asked many interesting questions. He gave many interesting answers. And I would like to continue my talk and what we are doing today in the light of what he have said at that time. When he would ask why he wanted to go for infectious disease, he said, because he was interested as a kid already, and he was fascinated by looking through the microscope, and he had read a book that was called War on Microbes. I think it was not in English as a kid, so I checked for French or for Dutch books. I don't know. You have to tell me later, Mark. It was Paul de Kruif's book, The Microbe Hunters, or it was from the Strijd tegen de micro-organismen. But there you got inspired. The question was, and you got involved into it, and you were interested in the research. But what research? And there you say something very interesting already. And that fascinated me also in our discussions later we had, that this fight on war against microbes is an old-fashioned way of it. And we have to look, and we heard that from Sylvain before, we have to look into it into a more ecological view. And the resistance is, and also from my point of view, a co-evolutionary effect on what we are doing together on this planet, microbes, Homo sapiens, and of course all other species, and adaptation mechanisms, not patho-adaptation, but it's the adaptation of survival. And I think that this together we saw in the times when we found that there's not only healthcare associated MRSA or community associated, but there were mixed clones that could both, and even coming from other species than Homo sapiens, and being able to jump forth and back, and many interesting things. And there to see that there is not only difference in the epidemiology of it and the resource and the source where it comes from, but also in the clinical implication, because there were no really outbreaks within humans in the hospitals, very few one. And there was another impact, another clinical impact. So it was not only important to know how the bug is called in clinical microbiology, but it was important what the bug was able to do with the patient. And there was a language that was able to subdivide, to substratify the same species into subtypes. And that changed epidemiology, because you had not only place, time, person, and species, but subtypes. And that gave a fifth dimension to the epidemiology we were dealing with. And later, you also said that it would become really a nosocomial problem. But you said also, it's not only the medical problem. MRSA showed that it is due to budget constraints, and it is because our health care systems are sick. In fact, MRSA is a symptom of ill health of the healthcare system in many places, always a diplomat. The hidden reasons, I call them. And that's true, because we know all this, and we have Herman in our virtual meeting. And if you look at that, and you take this statistic, you see that the Netherlands is the one here in this, with very few antibiotics per inhabitants, and Greece in these statistics with this country with most. But if you look into other questions, you see that the expenditure on pharmaceuticals at all, not only antibiotics, is by far per inhabitants larger in Greece than in the Netherlands. So it's maybe not that we have to ask our questions that Dutch use less antibiotics than Greeks. No, they just use less medicine at all. And Greeks use more. And the question is why? So if you use less medicine, you might probably also use less antibiotics. And the question is, how does it come? We don't really know. We can imagine. But if we look here, who prescribes the medicine and the antibiotics, and who has to disinfect their hands, because touching the patients very frequently, it is interesting that there are major differences. In Greece, you have very few hands to touch the patients, and they are running from one to the other and to disinfect and have to disinfect, to comparison to the Netherlands, where we have much more nurses per inhabitants. And on the other side, there are by far more doctors that can prescribe antibiotics in hospitals and practices in Greece and also in Italy than in the Netherlands. So that you can continuously say we have to do guidelines and explain to the doctors in the countries that prescribe a lot, don't do it. But who would write the guideline to tell to the system, why do you have so few hands for your patients? Or why do you have so many hands for prescribing? I think that are context-related questions, the hidden questions we have to look at. And that Marc foresees at that time. And the most striking is this one, because we all know that you see that in the Netherlands, 13% of all babies are born by sector cesarea per 1,000 live birthing. And it is in Greece and Cyprus and Italy about double as much, and even in Germany it's more. And we all know that there's also the question, they're giving antibiotics before, after the cut, but however, it will have influence even for infectious disease and antibiotic resistance. But I show this example because it shows you, well, what is the reason? Because we don't have to talk about antibiotics, we have to talk about the indication of why here women are operated, or healthy women are operated usually, and why is the difference so enormous between the countries? And that is not only explainable by medical reasons. And the next thing is that we look into our countries and we have to ask ourselves, is that true? Because if you see here the use of antibiotics in the Netherlands on a county level or on our neighbor state, which is Niedersachsen, you see that there's a nice, interesting phenomenon. The closer we come to the border here in the north, the more antibiotics are given. So it's a clear, for the statisticians here, a clear proof that borders are a risk factor for the use of antibiotics. We cannot explain it. We did studies, of course. We looked into also because people say, yeah, there's more morbidity there. People are go more to doctors, more unhappy people also in this region. More poor people. But we checked in a study that has been published also in the children and has not lived for so long to be so unhappy and so poor in the whole. They are poor, but so high morbidity, and also they get more antibiotics in the border region and on the German side more than on the Dutch side. And that is that Marc answered in one of the questions. There is a great need for well-controlled intervention studies to understand the diagnostics, how they can be used, and also our typing methods. And the first, of course, in what context should we do so? And I would like to invite you to go with me that we might ask ourselves, can we continue a national epidemiology? We see now in COVID that infectious disease start somewhere and they do not spread nationally. They spread in your context, in your region, and then the confluation between the regions become a nation. And this regional intervention reality is an important thing that I was inspired. I remember in early meetings with Marc and others, and from there to look into the regions. And there are many different activities in this region of about 40 million inhabitants, Netherlands, Northern Belgium, North Rhine-Westphalia, Niedersachsen. And there are many European funded projects in that. And one I would like to show you what comes out of that, because you see that here 23 hospitals by Corinna Glasner done a study across the border, and you see that there's difference, not anymore so much between MRSA left and right of the border. That has changed also in many other countries. But if you look into another microbes like VRE, you see enormous differences. And if you look into national statistics, you don't see them. And you have to ask yourself, so what is relevant for us to control it? And of course, we look into the regional one, but also cross-border. And it's nice to see here the young generation of colleagues. They then combine it and make, there is the Dutch side of the border, the German side of the border, and then there's a border region as a real entity and real epidemiological reality that has to be related to maybe the rest of the country. And what comes out here as an important message is that we usually thought that this is a problem of larger tertiary hospitals, but it's not. Also, the smaller hospitals, they have evenly they take part of the epidemiological pressure. And that's important because there the patients are going forth and back. Interesting was that sometimes in interviews you say things that later on we learn that they come out differently. And Marc said, unfortunately, I'm not convinced that we will still see that we will control MRSA at hospital level. Maybe we have not controlled it in Europe, but that's a masterpiece of that common effort really was able in most countries to reduce it, and in many countries also to reduce a lot. And but that's not a problem to not had the right idea in an interview about 16 years ago, because it was the best case scenario. It came out, but still we don't have it really under control. Interesting was that you said at that time that in your laboratory, in fact, you were developing molecular tools and rapid diagnostics for nosocomial settings. And that really inspired me. I remember we were in Prague in 2003, and I listened to that, and together with others, Herminia de Lancaster also, Alex from Belcom, Panayotis, so many people. There was this time of new methods, and there was a question of harmonization, and that was the prevalent of next generation technologies coming. And you see here, Sylvain described the IMEMs, and in the first you had the IMEM, and here it was in Switzerland, and you see Rich Goering, who's also in our meeting, on his knees in front of an important person, as we see from behind. And what was it? Because it was the next year in overseas, and Marc hands over, and thanks to Guido Werner, who's also in our meeting here from the Robert Koch Institute, gave me this picture, and we see Sylvain. It's always the same. Sylvain is always the same, and of course Rich and Marc also. And these meetings were very important, as Sylvain said. And without that, we have talked before, it fits very much as you introduced it, because at these meetings, these meetings were evolutionary pushers, evolution of technology, evolution of knowledge. And you see here in 1996 already, the first consensus guideline, first author Marc Struelens had been later in 2007, a renewed version by Alex from Belcom. And then came the SPA typing activities with Dag Hamsen, Alex Melma, and others. And there we were able, with very economic way, to do this 5-dimension epidemiology, molecular epidemiology. And then we had new questions that we could answer. And that led to really large studies. These large studies then firstly led by Hayo, Grundmann, and then taken over also for other microorganisms. And the discussions from Paris IMEM that Sylvain organized on, that there was this meeting. I remember that Marc organized the first brainstorm on typing. And I remember, as Andreas at the beginning, it was very difficult to enter with the technological world into the epidemiological world. But at the end, it came out, and enormous effort to get these technical reports done by the ECDC. Excellent. It helped to get it done at the ground, and was the wind in the sails for this technology. And now we're talking about metagenomics, but we're still in the whole genome sequencing typing with all different facets that are using on all different levels. And just to give you an example, some examples here, I don't want to go into details, how we're using it, that you really can, in the clinical setting, directly from the patient, get information, one-stop shop on the what do you find in here dengue viruses, and what subtype is it, and what is the reaction of the host if you do it on the metagenomic level, but how to use this technology on a regional level is what you can do then is a swarm prevention, and you generate unique marker genes in an outbreak, and then you give this information to your colleagues in the region. And then they can use these unique marker genes to identify by mass screening only the problem of the moment. And when the outbreak is over, you can throw the primers away. Maybe it comes back. You can have it in the freezer. But however, you don't see all the other problems running around at that moment. You can concentrate in the region on this single problem. The future, what will it bring here? One of our groups on microbiological imaging, Marleen van Oosten, that you can bind antibiotics here, vancomycin, to tracers, light tracers or radioactive tracers, and then you can really find in the imaging and also in situ the infections and treat them. and of course, a lot of developments that are coming and that we'll be using, especially in this kind of pandemics, will come a lot of a push for technology, and that can be used here. And if we look at that, the terahertz spectroscopy to identify viruses, now also COVID, but for the breath test, but also in microorganisms like Staph aureus. And there was a very interesting POCT webinar over a meeting organized by Marc and colleagues. So finally, and to come also to round up, next to the next generation of people, we have to go to the next generation of technology, to the next generation of people. And one of the important things is that here, in the tradition in ESCMID, with the sustain of many partners, especially also ECDC, we're trying to get the young generation to the next steps that we have to deal with, giving them European competence, because if I'm able to control MRSA in the Netherlands, I cross the border to Germany, or I go to Italy or Greece, I become incompetent or relatively incompetent, because it will just not work because in another context. And the same is true vice versa. And next to the European competence that we have to explain to them, what is the context you're working in, and not one guideline will fit for all regions and countries. The meta competence in being able to work together in transdisciplinary networks. That brings me to the last slides. I think that being inspired by the last 15 to 17 years working with Marc and colleagues, I think that in Europe we will be able to have these ideas of saying we can make the effort to make Europe free over 10 to 15 years of dangerous antibiotic resistance. But it will be an effort that is above all national or regional things, that we need to have first the idea to be born and then go for it. Because the approach will be dependent on the different healthcare systems. However, there will be one common approach. And it is a daily challenge in all healthcare institutions and regions that can stimulate and foster structures that will be helpful in moments of crisis, because nobody will give us really long-term structures for a crisis that comes every 10 to 20 years. There will be, but then it will stop again and say, yeah, but for once in 20 years, will we really invest? And you have to invest in infectious disease, and antimicro resistance is one of these daily infectious diseases that will foster structures we can also use for what we have now. Coming to the end, you said also something 15, 16 years ago about the ECDC, and you said, it may somehow start as an underfunded and understaffed agency, unfortunately. And in general terms, now speaking as a citizen, rather than a microbiologist, I think that it comes down to, do we really care and want to build health security for European people? Marc, I would like to tell you, you definitely did care. And I thank you for everything you did and contributed to the scientific and the public health and microbiology community. Thank you very much.
Thank you very much for a very inspirational and great talk, taking us backwards and forward with a clear view to the future, and we should go and paying tribute to Marc, and showing also the important disease we also see, particularly at the moment, which we should take as an opportunity to think about this in the future as well. I don't know, are there any questions in the chat? I can't see any. So again, I would like to pass on to Marc and see what question Marc would have for you. Marc, the floor is yours with a question for Alex.
Thank you, Ines. Well, I don't know what to say, Alex. This has been such an overwhelming talk, and thanks for all the warm words. And looking back at some of my silly answers to this interview many years ago, so that was a good one. I've forgotten some of that stuff, but it's good to look back a few years later. But I just want to answer on the detail for the anecdote that it was indeed Paul de Kruijf's The Microbe Hunters, which happened to be translated into French with The War on Microbes, but that was a wrong translation. So you spotted the right one. Very heroic, romantic vision of Pasteur and Koch, which really did inspire me as a kid. Anyway, so but no, rather than a question, I would really like to congratulate you on your concluding statement and how very much I see that as a not to be missed opportunity for all of us in the health community, public health, clinical level research is really not to miss this disaster we are facing now, as also as Andrea Ammon pointed out, as a revelation of all the structural weaknesses, how much we invest, how much we care for care specifically, and what you said, to look at where the problem happens, how we can partner to solve them, which is the local and the regional level first and foremost, and do it with complete community engagement and resolution, and not wait for disaster to try to pedal back. I mean, I very much appreciate and encourage you that this is the final word we should all emulate. Thank you so much for your vision. Over.
I think Daniel has a question.
Yeah, maybe this is a too big question, but Alex, what do we need to be CRE free in 2030? What's the magic recipe there?
I think the magic recipe is to focus and to have the idea of we will be able to control, and we will focus on that and not be afraid of this and saying it's not possible. It is possible, and then have using the network of public health and clinical networks that exist in all countries and regions, going down using technology where it is to help the ones who don't have it at the moment. So bring the water to the fire and not hold it for yourself. And being generous in that, and having, of course, stimulation, and then doing, for example, one idea could be by the unique genetic markers. What is the problem of your region at that time? And then focus on that very strain and try once to control at least one subtype, and not try to do all the jungle of microorganisms that you will never understand what is happening epidemiologically. So understand it must not be one species. Everybody, we will go for it, and everybody will control his problem of his region. I would love to be part of something like that.
Thank you very much. Daniel?
Yeah, there was some questions coming in here now on the chat, I think. Let's see.
No, I couldn't see any.
Couldn't see any?
No.
Oh, there they are.
Ah, there is one question. Sorry. Looking at the map regarding the spread of MDR in Europe in 2020-2013, clearly Scandinavian countries doing well compared with other EU countries, despite they get large amount of human influx in the last 5 years from the third world. How could they make the balance? Alex,
That's an interesting
ideas?
question, and the answer has been given in the interview by Marc, because there was a question, I haven't shown it, where exactly that was shown, because at that time there was the opening of Eastern Europe that had influence, or that was supposed to have influence to Western Europe, and it was not as foreseen. And the same will be true here, because we see that the influx of people is not the people with the chronic disease, and so it's totally different. A lot has been written and talked about. I think migration has always been part of human society, and it will not be different. We just have to focus on what is important when all these people that migrate or not are becoming a patient. So the conversion from citizen to patient, and then they're entering the system where they can be harmed. And we have to look into the ecological part to make the system patient safety for them, and maintain the prevalence of resistant clones that can transmit in epidemic waves throughout regions and countries and over countries. So that is where you have to focus on, and that starts, you have a global view or a European-wide view, but it starts somewhere where the small outbreaks are that can become larger epidemics.
I see we have one more question from Guido Werner. Why do hand hygiene and antimicrobials work for MRSA that well, but not for other MDR pathogens like carbapenem-resistant, Enterobacteriaceae, I guess it will be? Klebsiella he has. Okay.
Yes. Yeah, that's the question, and of course it is in a world of microorganisms that are the MRSA world. Let's say first, the transmission is not so relevant via the fomites and fecal orally. In a world of fecal oral transmission that you see as VRE or also gram negatives that are gut bacteria. You have totally different transmission dynamics in hospitals. Imagine all the mattresses where people come on, and if they're not disinfected, or if they cannot be disinfected, you can imagine that we are stamping the microorganisms from one patient to another. That's only one example. So much more complex. If these microorganisms that are used to survive in the environment. And in these highly dynamic environments, you see that they need other approaches. So it's a combination of hand hygiene, environmental infection control measures, but also if you do all, and the reduction of selection pressure for prevention of resistance, but if you do so still, the most efficient way of transmission, and we don't have that in mind usually, but the most efficient and the most sure transmission way is that the microorganism follows the host, and in the nose, in the throat, in the gut, and the hosts are continuously moved from one ward to the other, from one hospital to the other, to the nursing home and back, and we cannot avoid this transmission because we cannot stop the patient. But if we don't look into the system as it is continuously moving, and we move them by merging hospitals, by sending them away, and that is the transmission that we usually overlook, and we look into R-naughts in hospitals, but we never look into R-naughts in regions, not so frequently, and we should, from my point of view. Other interventions are then necessary.
Okay, with this, I would like to thank you very much, and we're a bit behind schedule, so we just decided that we wouldn't do a break, and thank you very much, Alex, for this great talk, and I pass on to Daniel.
Thank you, Alex.
Thank you, Alex.
The next speaker. Let's see if
we need someone to turn off the mic.
Can we try again? Okay, no, I think it works. No. It was a long delay there. Okay. I don't think this works. We need to wait.
Okay.
I try again. Can you please look at your microphones? And turn off if you are not speaking. Okay, I try again here to see.
And the room locked to users. Maybe that's why.
and then leave it.
Hello?
Okay, we try again.
Okay, we try again. Hello, can you hear me?
Yes, Herman.
Yes.
Yes. Can you hear me?
Yeah.
There's
Hello?
still a bit of echo, but I think we need to continue here.
Nej, det kan inte.
I would leave her.
Hello, can you hear me?
Yeah. Yeah.
Hello?
Yes, Herman, we can hear you.
Oh, sorry. Sorry. Can you present the slides maybe? Is that possible?
Yeah, we will make a try here. Sorry for the
Okay.
technical problem.
Oh yeah.
Is there a problem on my side or is it on your side?
I think the problem is on our side. We have an echo.
Okay. Okay, so they can hear me without an echo apparently.
Herman, did you send me your slides?
Yes,
Over?
yes.
Over.
Yes, I did. I sent them around 2 o'clock.
Okay, I have them, Herman. I think I will just leave the floor to you, and then we will fix the echo. And we will do the introduction of you later on.
Sure, no problem.
Okay.
That's okay. Yeah, absolutely. Okay. Yeah.
Please, Herman. Sorry.
Yeah. Sorry. Okay. Thank you very much. And I'll see if I can move the slides forward, but that should work. If not, otherwise I'll ask you to move the slides forward if there's a problem. Good afternoon, everybody, and good morning to our friends and colleagues in the United States. Of course, I wish we were all together in the same room, but unfortunately that's not possible, and I'm very unfortunate indeed. But I'm very honored to be nominated by Marc for his retirement symposium. It's obviously something you cannot refuse, and again, it's a really great honor, Marc, that you have invited me for your final retirement symposium. I usually agree with everything that Andrea says or that Andrea does, but today I cannot agree with her, because she said in her opening speech that she was very happy that Marc had chosen ECDC to continue his professional career. And I have to say, Andrea, I cannot agree with you, because for us it was really a great loss that Marc left Belgium. And I can see that Frederik Jacobs is also online, and I'm sure that Frederik will agree with me that it was a great loss for Belgium. It was great for ECDC that Marc joined this fantastic institute, but we lost a great colleague, a great scientist, and a great microbiologist. So, Marc, you've asked me to talk about translating medical sciences and also about pandemic preparedness and response, and that's something that you and I have been working on for the last 10 years, and you have been giving us a lot of advice, but we've known each other for a much longer time. And I was thinking when I was making my slides yesterday, when we met for the first time, and I think it was in 1981, because we were trained in microbiology in the same group of Jean-Paul Butler. And I think that's where we met for the first time. And then you left, of course, for Bangladesh in the ICD-DRB, where you did your diarrheal disease research. And I continue also to work on diarrheal disease. And then you came back and then you moved to Erasmus Hospital. So you continued your career in Erasmus, and then eventually you moved to Stockholm and did fantastic work, as been shown today. And there was a photo of Richard Goering with Marc, and Richard being on his knees for Marc. And I think I know why he did that, because many years back, I recall that Richard told me that the typing work of Marc was outstanding, and he said to me, Herman, this is the best typing work that I've seen so far. This is really outstanding. So not surprised that he was on his knees on this photo for Marc. Now, Marc has not only been a world leader in antimicrobial resistance, but he's also been a very visionary person also with regards to the coronavirus. And I will prove to you later that Marc already thought about corona as becoming the virus that could call the next pandemic. And he might be even surprised himself, what I will show him later. But it just shows how visionary he was. Okay, so that's just an introduction, and I will now continue with my presentation, but I will not discuss all the slides for the sake of time, and I guess the slides will be shared afterwards, and I will just show you what we are trying to achieve in the European Union. And I hope there's not an echo, because I see some messages here. Is there an echo in my voice? Is that okay?
Your voice is fine.
Your voice is fine, Herman.
Okay, good. So, could you maybe give me the next slides? Is that possible? Yeah, thank you. So I wanted to start with a quote from Mike Osterholm in his book Deadliest Enemy, and I often use that quote because it talks about the pandemic threats, and he's really saying that there are two pandemic threats. One is influenza, and the other one is antimicrobial resistance, and both are pandemic infectious diseases. And I started working on antimicrobial resistance, and then moved into respiratory tract infections, particularly to viral infections. And I will show you what we're trying to achieve in Europe by bringing these two threats together. If I have the next slide. The next, yeah. So basically what we're trying to do is to leverage EU investments in clinical research and antimicrobial resistance, and indeed on emerging infectious diseases. And I will briefly discuss a few projects and then try to explain what the long-term vision is. And it's probably the reason why Marc asked me to talk about this, because I know he's very supportive of this. He's been an advisor to the Prepare project and gave a lot of outstanding advice during our scientific advisory board meetings. And I guess, Marc, it's probably the reason why you've asked me to talk about this. So I'll briefly discuss Compact and Prepare. Compact is an IMI-funded project. Prepare is a project funded by DT Research. I will not discuss Value X. It's another exciting project funded again through IMI. Briefly discuss Recover, and then of course Equate Plan, because Equate Plan is really the business plan that we're writing for the moment for the long-term future of clinical research in infectious diseases. Next slide, please. So just one slide on Compact. Compact is really, as most of you know, it's all about combating bacterial resistance in Europe, funded through IMI. Mark Bonton is the coordinator of Compact, and there are several Compact projects: Compact Net, Compact Care, Compact Magnet, Compact CDI, and I see many of you online today who are actively participating in this project and who are very supportive of this. And Compact is, if I have the next slide, is really a collaboration between the academia and the private sector, and it's built on 4 pillars: ClinNet, LabNet, StatNet, and EpiNet, and I will explain that later. Next slide, please. So the next project I really briefly would like to discuss is Prepare. That's an acronym for Platform for European Preparedness Against Re-Emerging Epidemics. It's again EU-funded project, started in 2014. It runs till January next year, and is coordinated by Mendo de Jong and myself. And I think the easiest way to explain Prepare is just to read to you our mission. And so our mission is to establish Prepare as the European clinical research framework for large-scale clinical research studies on infectious diseases prepared to rapidly respond to an emerging infectious disease, and then with basically two objectives. One is to provide real-time evidence for clinical management of patients to make sure that we do a better job than we've done in the past with other epidemics or pandemics, and then, of course, also particularly important today for informing public health responses. Next slide, please. If you can click again. And again. Yeah, so this is the whole idea of and it's interesting actually, I made these slides in 2013 when we started Prepare, and when I was looking at these slides again yesterday, I was really wondering to what extent we were right when we were trying to explain what the issues have been so far. And the issue, of course, is that if you have a pandemic situation and we've just finished the first wave, you're waiting for the second wave, you have a very good public health response, you have a very good preclinical research response, and I'll come back to this, and clinical research is lagging behind. So the next slide. So the whole idea in Prepare is to move, if they have the next slide, you can click again. Next slide, please. And again, and again. Yeah, so the whole idea now in Prepare is to move fast forward and start clinical research much faster. And so I never thought that at the end of Prepare, I would be part of this huge pandemic, and who would, of course, have thought that. And I never thought I basically would become a full-time virologist at the end of my career, but that's what it is. And so the question now is, did we really respond to the COVID-19, and did we rapidly respond and conducting the clinical research that we needed to do? It's an interesting question, and I'll come back to this. Next slide, please. So in Prepare, we're doing a lot of what we call peacetime studies, observational studies, and adaptive platform design studies. Many of them have been completed. One particular one, the REMAP-CAP, is a randomized embedded multifactorial adaptive platform trial for CAP that has been converted, if you like, into a COVID-19 trial. And so that is the whole idea. And that was the whole idea when we started Prepare. We start with adaptive platform trials. We give it the opportunity, the possibility to adapt the platform and to add new arms if this is pandemic infectious diseases. And so we've done that indeed, and have changed REMAP-CAP into a REMAP COVID adaptive platform trial. And we hope to sign a contract with the European Commission next week of another 15 million, where we would like to include more patients in, if we have a second wave, in this REMAP COVID trial that was originally the REMAP-CAP trial. So next slide. Now, we discussed a lot, how do we respond to an emerging infectious disease? And we developed 4 modes for that. So mode 0 is basically the peacetime trials, like the REMAP-CAP trial, if you like. And then we decided that we needed 3 modes. And the first mode would be if there's a limited threat of an infectious disease, that we would move to mode 1, where we start Prepare. We start thinking about, let's say, the resource gaps, the research gaps, and we start writing the protocols, where we keep the protocols, let's say, on the shelf. We don't mobilize. Mobilize we only do when we see a potential threat of infectious disease, and then we roll out the protocols. We get approval of the protocol, but we don't. So we plan the trial, but we don't really start the trial. And then finally, if we have an immediate threat, and that's what we experienced, of course, in January, February, when you have an immediate threat, then we push the button and we start implementing clinical research. And that, again, that's what we did. So next slide. So then, of course, came January 2020, and the first paper in New England Journal reporting on the novel coronavirus from patients with pneumonia in China 2019, and then many congratulations to Eurosurveillance because they published very quickly our paper led by Christian Drosten on the PCR test, and that could have made an even bigger difference in the European Union if countries had prepared themselves better in terms of rapidly responding and rolling out their rapid diagnostic tests. And I think only Germany has been successfully doing this in February. But I think, unfortunately, although the evidence was there, the paper was published, unfortunately, most European countries did not respond rapidly enough to roll out their rapid diagnostic tests. Next slide. Now, so Marc Struelens, and I'm coming back to what I said at the beginning, that he's a very visionary person. Because Marc sent me this email on the 3rd of December 2012. Headings are in Flemish, but the email was all about a novel coronavirus. And this is not COVID-19, and of course not, but he was asking me information about another coronavirus, the Human Coronavirus Emc. It's a virus that was causing deadly disease in the Middle East. It appeared to have originated from bats, and then it turned out later that it was not the same as the SARS coronavirus. So Mark was concerned about this. He was concerned that coronavirus could cause a next pandemic, and I kept this email, and I couldn't believe myself when I found it yesterday, Mark. But I kept this email, and actually I remember when I received it that I was thinking, my goodness, Mark is becoming a virologist now. He's moving from EMR to virology, which is quite an interesting change in his career. But as I said, most of us have become virologists over the last 6 or 7 months or so. So next slide. So we submitted funding, a request for funding, or at least a grant application to the European Commission, DG Research, and it's amazing how rapidly the Commission responded to this outbreak. I think they were one of the first to fund clinical research. They were one of the first to indeed allocate funding to specific grants and specific projects. We were very fortunate, and we received 5 million euros from the European Commission to study the SARS corona outbreak. Next slide. So this is what we basically did. We moved on the 22nd of January 2020 to mode 1, and I explained to you earlier what mode 1 means. And then very quickly, and I think we really did a fantastic job by rapidly responding to this emerging infectious disease. We already on the 6th of February moved to mode 2, and then on the 2nd of March we decided to push the button and start clinical research. And that was before a lot of countries started the lockdown. There was a lot of debate also in my country about the fact that this is just a simple flu, and what are we worried about this? It's just a virus that will come and disappear, just as the SARS coronavirus. And so we did start our clinical research, and I can tell you it's not that easy, because if you want to start clinical research, you start investing a lot of money, and you may take a big risk, because you need to mobilize sites, you need to ask them to participate in trials, and some of them were saying, what the heck are you worried about? This is just a simple flu, so why should we do this kind of research? Next slide. So I will not dwell on the research that we've done so far, but I just want to briefly mention the lessons that we've learned. If I have the next slide. So what we did was we did qualitative research, if you like, and interviewed about 40 experts in Europe, just to see what the learnings are from the research response in Europe, what were the positive learnings, and what were the negative learnings with regard to clinical research response. Next slide. So what came out of these interviews with these 40 people was, and I'm sure that the colleagues working for European agencies will be happy to hear so, that funding and collaboration with ECDC was easy and really very very rapid. Fantastic communication with the European Commission, flexible and pragmatic response, high interest from the EC and ECDC in the project. Quick availability of funding and also rapid budget approval. So that is something, and I think maybe not enough highlighted, but this is something that the European Commission should be proud of, because they have responded very rapidly. Also, sense of urgency, and I'm sure you've all experienced this, and willingness to collaborate. Willingness of the medical community to collaborate, and the great sense of the community indeed to collaborate. And I think, in retrospect, if you look now at what's been done over the last 7 months, I think this is a victory of science. It's really amazing how much science has been published. Not always fantastic science, and you can talk to Lancet what they think about this, but certainly fantastic papers have been published. But we have failed in another thing, and I'll come back to this. But the other thing, what really astonished me was the ethical approvals of the member states. We had spent 5 years in prepare to see how we could deal with these ethical, administrative, regulatory, and logistical barriers, the old barriers. Basically, it was a 5 years of waste of time, because countries really responded very fast, and the ethical approvals were done within days, and that I had certainly not expected. Next slide. But there are also negative learnings, and I think I just want to previous slide, please. Yeah, I think the negative learning is that clinical research has failed. It has failed globally, and I think it has failed in Europe as well. I think it could have done much better. I think there are very good reasons why it has failed. I think there are many lessons to be learned from this. It's not always because of the lack of preparedness and prepare. I have many reasons why I think we have not succeeded as we should have done. I don't want to really just dwell on this tonight or this afternoon, but certainly I think globally, clinical research has failed. And every physician who saw 5 patients with COVID-19 decided he should do a protocol. And I've seen this in hospitals in Belgium, I've seen this all over. You see massive amounts of protocols, too few patients and too many studies. And that's also shown on the next slide. Next slide, please. So this is just a slide from R&D blueprint summarizing the RCTs, and this is an under let's say calculation of the number of studies that have registered, and look how many have completed. That only 15, and so most of the studies have not been successful. And I should say that one country that has been very successful is the United Kingdom, and it's amazing what they have been able to achieve, and there are many lessons to be learned from that as well. So next slide, please. So what we really, and I'll finish now with some slides on the future. So what we really would like to do now is to build a European Clinical Research Alliance in infectious diseases. And that is by combining what we have done and invested in combat and in prepare. So the next slide. We've done a lot of thinking the last 4 or 5 years, and again, Marc has been part of some of these discussions about what should be the purpose. And again, I will go quickly through these slides. So the purpose is to reduce impact of infectious diseases, both at individual and the population health. On the next slide. The vision is to, and again, I read this very quickly to you, it's efficiently generate the rigorous evidence for new, but also for improved both diagnosis, or let's say for diagnosis, for prevention, but also treatment of infectious diseases threats. And then again, and very importantly, to facilitate European multidisciplinary clinical research network and innovative research approaches. Next slide. So we spend a lot of time discussing the values. And again, you have the slides afterwards, and there's no time to do this or to spend time on this. But I can tell you, we really thought very hard about what should be the values of this European Clinical Research Organization. And we felt that scientific independence should be at the core of eCRADE. And then, of course, also it should be excellent, not for profit. We don't want we want a not-for-profit organization, total transparency, responsiveness, sharing credits and benefits, and that's, I think, what we're good at in Europe, and also a very efficient organization. Next slide. So this is the strategic agenda. Again, no time to discuss, but we want a sustainable clinical research network built on past successes, and I've tried to explain this, what we've done so far and how we want to build this. Financially sustainable. Eventually, we want to make sure that there's enough funding to keep it sustainable, but both support from the public and the private sector. We don't want to focus only on Europe. Of course, it's a European organization that we want to build, but we want, of course, to be globally connected, build a community of networks, and we also want to encompass all aspects of clinical trials, then generate evidence to improve care, of course, and then finally, we want to be innovative and flexible, and I'll show you some of the innovativeness that we would like to achieve. Next slide. So really, we've been spending a lot of time, efforts, and money on building what we call a warm-based clinical research network. And that includes, first of all, primary care sites. And for some of you familiar with GRACE, you know that we've done huge amounts of studies in between 2006 and 2011 in patients with community-acquired lower respiratory tract infections, and built a very solid network of GPs led by Chris Bertler in Oxford and Theo Verhey in Utrecht in the Netherlands. Evelina Taconelli has been building a network of long-term care facilities. Marc Bronton has been building a network of hospital sites in more than 42 European countries. Carlo Giaquinto already in the 1990s started building a network of pediatric sites, and I've been trying to build a network of 800 labs in more than 40 countries. Next slide. So this is the infrastructure. Basically, it shows in a different way. We have a clinical research infrastructure with pediatrics, with hospitals, primary care, and long-term care. We have a lab infrastructure, biobank infrastructure, and we're discussing now the business model of such a biobank, which is not an easy discussion. We have EpiNet, led by Evelina Taconelli. We have StatNet, led by Stefan Harbert. And so this is basically the structure that we'd like to leverage for future infrastructure for clinical research in infectious diseases. The next slide. So the next slide shows you the full eQuate package. Again, no time to discuss the clinical research package that we would like to offer, would like to do the lab research network, AP research, statistical research, data management, biobanking, and then finally, of course, training and patient management. The next slide, please. We had a lot of discussions about innovative design that we would like to build into these clinical studies. And so basically, these are the kind of studies that we'd like to do from let's say, the continuous enrollment of patients in the perpetual observational studies through studies with 2 control groups, to response adaptive randomization studies. And you've seen that we've done this already to some extent in BMAP-CAP, to studies with master protocols, nesting clinical trials, and then again, platform designs, studies. So those are the kind of trial designs that we would like to start in the future. So the next slide shows you the different design elements in our studies. In green, you see the ad hoc studies that we would like to do with industry and also driven by academia, and the types of designs of the different studies, randomized controlled trials, of course, RCTs with controls group, etc. And then in blue, you see what we would like to do, the various observational studies, again with the different trial designs, and that's what we would like to do now, and hopefully the next 5 years, to start these perpetual observational studies, and then industry, for instance, can plug in these worm-based trials. The next slide. So the value that we offer here is really around reducing time, reducing costs, and also providing high quality research. And in Comeback, we developed key performance metrics to measure these values. And again, I won't discuss them. Next slide. So we also estimated the time that is gained in completing these clinical trials, and that we've done with Lloyd, because as I said, we have received funding from the European Commission to build the business plan, to write the business plan. We do this, and that is the eQuate Plan project. We're doing this together with Deloitte, and for instance, they have helped us to calculate what are the different gains in the different phases of the clinical trials. And we've estimated that this should be at 28%, or about 7 months gaining in these clinical trials. Next slide. So this is the organization that we would like to establish in Europe. The idea is that we build eQuate with the Netherlands and Utrecht particularly as the headquarter. That would be the coordinating center, would be the country, would need to be the coordinating center, where the governance structure would to some extent be based. And this would be based on with the Dutch foundation under the Dutch law that needs to be established. But then, of course, typically Europe, we don't want a centralized system. We want a decentralized system with co-location offices. And these co-location offices are basically built on what has been done over the last 10 years, built on LabNet, Epinet, StatNet, and all the other networks that we have involved so far. Next slide. So that is the governance structure. Again, I won't discuss it in detail, of course. So that is the governance structure that we'd like to establish with the General Assembly. And the General Assembly is basically the clinical sites, because they should own this infrastructure. They are the most precious part, if you like, of this organization. And then further board of directors with several organizations that would be a part of this larger governance structure that we are currently discussion has not been finalized yet, and needs to be finalized very soon. Next slide. So the next steps, we have a meeting, as I said, actually we have a meeting next week with the eQuate Plan Consortium, where we will review the fourth version of the eQuate business plan. We should finish this by the end of this year. We have also submitted a proposal where we hope to get funding to do the observational study. So hopefully we will find out in October, November if we get funding for this. And then we prepare an eQuate plan. Normally the end of this year, early next year, we were supposed to have a very big event with more than 400 people, but that we had to cancel. So it's going to be a much smaller and probably a virtual meeting, unfortunately. And that is, of course, sad, but that's the way it is. We hope we probably extend eQuate base because of the time that we lost due to the pandemic until the summer of next year, which gives us time to establish entity and to detail the operational aspects. And then we hope to go live with eQuate mid 2021 if all goes well. Next slide. So this is the whole consortium. You see many of the names, and some of you are participating today, and very, very grateful to all the support that we have received from many of you to try to establish this organization. The next slide. I listened yesterday to the speech of the State of the Union address of President Ursula von der Leyen, and I was really very impressed. Usually these speeches are very boring. They say nothing basically. But I was very impressed with what Ursula von der Leyen was saying yesterday. She stood out, made some very firm commitments, was very passionate. It's something that's often been missing in the past. And again, my admiration for what she did yesterday. And she particularly mentioned, of course, health, particularly, and I was very pleased to hear that, the fact that we need to build a stronger European health union. And I like this very much because there's been a lot of emphasis on the monetary union, on money and in economy, and European citizens felt that they were not really connecting to the European Union. But that's because the member states didn't basically allow the European Union to do so. So I hope that we will indeed be able to build a stronger European health union. Great news, of course, for ECDC and EMA. They will hopefully get more support, more funding, and more staff, and they definitely need this, and they deserve it. And something that really surprised me, the European BARDA. I don't know exactly what the plan is here, but I was, of course, very happy to see this. So, final slide. So, conclusion, I think the SARS outbreak in 2002, 2003 created indeed this threat that was real enough for the EU to create this new agency, ECDC. So let us hope indeed that this pandemic is a wake-up call to build this European Health Union, and that there's more solidarity between the member states. And I'm actually very optimistic. I think this was a great lesson that we need this kind of solidarity. And maybe why not, maybe in 5 or 10 years, there's another new agency called eCRATE, and I would certainly be very happy to see this. So my last words are, of course, for Marc. And Marc, I would like to thank you very, very much for a wonderful and a very inspirational collaboration, but also for sharing your friendship with me. It's been a great pleasure to share this friendship with you, and it's been an honor to have had the chance to work with you, particularly in Belgium, less when you were at ECDC, but particularly in Belgium. So many thanks for your contribution to science, many thanks for your contribution to public health, and I would agree with what I think with what was said earlier by Alex. I think that Marc, yes, you did care about people, you did care about people's health, and it was great that you went to ECDC. And again, thank you very much for everything. And my last word just to say that please take care. Looking forward to see you in your house in Stockholm or in our house in Belgium, Marc. Thank you.
Thank you very much for the impressive talk, Herman. May I ask, Herman, if you could close your microphone while I take the question here. Okay. Oh, no, I think
Daniel, there was an echo in the beginning. We were not able to hear you clearly. Can you repeat your message?
Yeah. Thank you very much, Herman, for an impressive talk. I think we had some problems with the audio in the beginning, but I now think that Herman has introduced himself. I think maybe just to add is that he was one of the funders of the European Surveillance and Antimicrobial Resistance project that became EARSnet under ECDC in 2009, and he was also the funding coordinator of European Surveillance of Antibiotic Consumption Project, ESAC, that moved into ECDC in 2011. So, Marc, for sake of time, just if you could give maybe one short comment here, and then we unfortunately need to move further in the agenda.
Yes, certainly. I'm happy to do so, Daniel. Well, Herman, what to say? 1,000 thanks really for your talk and for your very warm words. It's been a shared pleasure and privilege, as you know, to develop our careers alongside first in Belgium, but also in your European Union initiative. And like everyone, I think I'm very impressed by the ability that you have again shown us today to transform the vision of what is needed in Europe into building a lasting institution to address those needs and building this really comprehensive clinical research organization, which which really will fly high under the current political support that we get from the highest level in the EU Commission, as you indicated. Maybe a quick question for you, because it's a bit of contrast of your negative conclusion on the quality and real life delivery of clinical trials for the COVID-19 treatment or diagnostics development, and what you expect with Ecology. So what would be the selling argument that really would be the magic bullet to overcome this competition that we have seen leading to fragmentation and ineffectiveness of clinical trials for COVID? Can you give us, in the big organization, what will be the enabling factor, in your opinion? Herman,
Can you hear me? Yeah, sorry. Can you hear me
Yes.
now?
Yes.
Okay, sorry. Yeah, thank you for that question, Mark. That's not a trivial question. I think indeed lessons to be learned here. What we did experience was that all the sites started their local trials. If I look at my own hospital, I think there were 20 or 30 trials at some point in time. And then of course, Stefan Harber gave a fantastic, let's say, explanation for this. He called this academic cannibalism. And he said he's never seen such a thing like this. And I think it's this true. Everybody wanted to do a trial. And then, of course, we came with our European-wide trials. And people were saying, yeah, but this is, we can't really connect to this. So this is too far away from us. We don't really feel ownership of this, and so that's a lesson to be learned. And yeah, we still would like to do our own trials. And then, of course, turned out that most of these trials will not have succeeded in anything. So I think that we need to connect now to these sites. We need to learn the lesson from what the UK has done, and they were certainly better prepared. They had the infrastructure already in place to do clinical research. I mean, they failed in other things, let's be fair, but certainly they had an infrastructure for clinical research that was more advanced and then the infrastructure that we have available. And so I think we were not ready enough. We need to go beyond this, but we need to connect more locally with the hospitals, with the ministries of health. That's another lesson we've learned. I have been saying, as you know very well, Mark, that we will fail if we cannot connect to public health. If we cannot fill the gap between public health and between clinical research, we will fail each time and over again. And I think that is, for me, one of the most important lessons. If we cannot find a way to bring clinical research and public health closer together, the next pandemic, we will fail again. Thank you.
Thank you very much,
Thank
Herman.
you very much, Herman.
Herman, if you could turn off your microphone, please. Okay. We come now to a very pleasant part for myself. I have the honor to introduce the reason why that brought us together, namely Marc and his achievements, his long career, his influence and his positive inspiration, as we heard from everyone. He's inspired all of us, and I'm sure he's been a mentor, not just for myself, but for many of us. And he's never had a formal role with Eurosurveillance, but he was truly a great supporter for the journal, and he was a speaker on our scientific seminar, and now I was very pleased to have the pleasure to moderate, as he did moderate for us before. And Marc, I don't think I have to introduce you. Please start your talk.
Marc, you're muted.
Sorry, can you hear me now?
Yes, now we can hear you, Mark.
Okay, can you see my slides as well? Or?
Yes.
Yes, we can. Thank you.
Very good. Something went wrong. I've lost my slide now. Okay, here we go. Really, I'm really overwhelmed today. It's been a really tremendous pleasure and honor to be invited to my own retirement seminar. It's fantastic to see so many of our colleagues and friends participating from around the globe, and I'd like really to thank you, Daniel, Ines, Andrea, for organizing it. It's been really a tremendous opportunity. And I understand this has been quite a long symposium altogether, and these virtual communication circumstances. So I like to conclude it by a little personal view on perhaps how I've learned along the way of my journey across medicine and public health, and learning about the fascinating complexity of the infectious disease challenges. And it's been tremendous to hear the three fantastic talks by Sylvain, Alex, and Herman to really set the scene of what the last 20 or more years have achieved and where we stand. So the point is, how have we moved? Are we moving from a more traditional to a more evolutionary approach to medicine and public health? Trying to advance my slide from this side. Yes. So unfortunately, traditionally, medicine and public health have been quite separate organizations in terms of people and mechanism of delivering services. Even though, as indicated in this little graphic on the left side from the US CDC, the advance of genomics and molecular methods has provided exquisite precision, both for medicine and for public health, and tracking which cancer or which particular pathogen is causing disease. In fact, the competition between the two sectors has been traditionally in our countries to the detriment of the population protection and focusing, as we know, most of the resources of a 99 to 95% of workforce and monetary infrastructure and funding to ever increasing sophisticated personalized medicine as opposed to long-term health protection of population. And I think that has left us a bit off guard when we were confronted with this disaster of COVID-19. There's a prehistory to ECDC, which has already been alluded to, I think, by Alex with this TLID interview back then. Before ECDC existed, I had a dream of an ECDC together with some fellow microbiologists and molecular geneticists, especially Michel Thiberac from Montpellier was in the lead in creating a new journal called Infection, Genetic and Evolution. And in an editorial together with Ferretti and Mascoma, we really were exposing our plea for the establishment of a European Center for Infectious Disease. We didn't call it prevention and control, as the response of the EU to address the major infectious threats being man-made or natural. And in contrast to the reality of what was established a few years later at the ECDC, the ECID activities that we were really advocating were, of course, the surveillance risk assessment training, but also advanced research, having a big biomedical research center as part of the center, a bit after the model of Institut Pasteur or US CDC. And with the idea that that would provide essential interaction on a daily basis between epidemiologists, public health scientists, and biomedical scientists, and cross-fertilization, as well as providing the multidisciplinary critical mass required for launching the holistic research on genomic and protein data flow that was started to become massively produced, and we're talking 20 years ago, and we see the reality of that today, of course, blooming. And I think that concept that we advocated at the time, among perhaps more the biological side of the game, has been nicely coined in a recent concept paper in the Lancet last year by Anderson et al. Under the term of microbial evolutionary medicine, which pleads for an interdisciplinary approach, again, that applies evolutionary biology to address challenges of and identify the root cause and determinants of infectious disease, and proposing the view that infectious disease is sort of the consequence of maladaptation of the host to the environment. We can see some resonating theme here. And they provide in that paper, which is worth reading, a little quite complex scheme that tries to bring together new tools, provide a new kind of data, and how this information flow can integrate to improve both our knowledge and our countermeasures for dealing with infectious disease. So it starts with the little test tube there with sample from the black person, the nil patient or population with disease or its connected environment. And of course, traditional microbiology way would be to look at the phenotype or drug susceptibility, but nowadays we can quickly generate full genome or large part of the full genome data. And these, especially on the collective accumulation of data, can be combined into database, pheno and genotypic data, and provide comparative genomics connected with the descriptive data from the population disease and exposure characteristic in epidemiological database. And this synthesize allow to provide and generate hypotheses that can lead to innovative treatment or prevention strategies. Importantly, the cycle should be integrating case-by-case hypothesis verification step through experimental research, being evolutionary studies, ecology studies, pathogenesis studies. And in fact, if we look at how this would apply to an increased understanding with advanced metagenomics and other tools, first of our internal microcosm determinant of disease and the advance in understanding the functions and composition of human microbiome, it gives a clear picture how we can switch from health, the normal gut, for instance, microbial homeostasis and balance, to the imbalance, the dysbiosis it's been called these days. And on the left, you've kind of a schematic overview on how the intestinal mucosa is surmounted by the outer mucus layer, which hosts vast communities with more bacterial cells than our own cells, so about the same number of hundreds of different taxa that interact with the food, diet, and themselves to actually maintain the integrity of the epithelium and regulate the mucosal associated lymphoid and immune tissue. So changing the diet or changing composition of this microorganism can disrupt completely the physical barrier and the physiological protection of the gut to the outside world. And that's what an aggression like antibiotics can do by wiping out a large part of this normal microbiota and opening the door to exposure and acquisition of multi-drug resistant organisms that can overgrow and further disseminate from that colonized patient to the population, as well as expose the patient through a disrupted barrier to translocation to the bloodstream and sepsis by this resistant organism, especially if they acquire, like the dual risk Klebsiella, additional selective advantage of capturing genes for hypervalence. If we now move from the inside phase of our interaction between microbe and health and the global environment, and we know we are in the planetary health alert phases, which can be seen in a way as a global dysbiosis as well. And not only are all the indicators in red alert with exponential growth, pollution, CO2, fertilizer use, chemical use, pesticides, antibiotics, but also we know that there is a tremendous reactive diversity of living organisms on Earth that is changing under this disruptive pressure. And of course, the polar bear is just sitting on the tip of the iceberg, but mammals are just a very small fraction of the biomass. And this is from a recent kind of estimate of the distribution by gigaton of carbon of the living matter on Earth. And you can see that bacteria plus fungi outnumber animals by over 50 times. So all these unicellular or relatively fast replicating microbes have this property, as was very nicely described again by our speakers today, to evolve much faster than we biologically, with replication time of minutes or hours instead of decades, as well as, especially for the bacteria, but interacting with viruses, to be functionally working like a single superorganism. And this is due to this very fast ability to transfer genetic information to the so-called horizontal transfer mechanism that we have seen at play for the rapid evolution of microbial pathogen in Sylvain's talk. So going back to reality, CDC was created in 2005. And I joined in 2010, and I was offered the privilege and task to help address the legal mandate for microbiology, which was not to do advanced research, but to foster cooperation between expert and reference laboratories and foster the development of sufficient capacity across the EU for essentially diagnosis, detection, and identification of agents that threaten public health. Mind you, it didn't foresee that we would have to ensure that microbiology can also provide support to the response itself to the threat should it develop into an epidemic or a pandemic. So how did we start? We started discussing for a couple of years with our national expert for national microfocal points and the member of EU networks that would first need to understand what the capacity is made of. And we came up with this concept of virtual EU public health microbiology system, which in fact is grown organically by the aggregation of 30 very diverse national systems with national organization, historical tradition, and resource level. But by and large, they all do in every country. First and foremost, not just the expert and reference lab, but the clinical labs, or they do the bulk of the diagnosis, drug susceptibility testing, and screening services. Then on top of that, in each country are the national reference lab who serve certain public health function, like investing in outbreaks, laboratory-based surveillance, and advanced identification or characterization of rare or dangerous agent. And this is wrapped together with interaction on all three levels by networking and sharing data, sharing expertise, and promoting method quality and harmonization where needed. And we came up with a set of capability indicators that cover these three layers: the primary diagnostic to the left, the reference lab service at national level in the middle, and also the surveillance, but also response support function of laboratories. This is across a number of specific targets with bunch of indicators, either infrastructure, process, or outcome that measure the capability and capacity of these services across each country. So as Andrea said, we were able, by having 100% participation over the last 5 surveys that have been conducted so far, and thanks greatly to Katrin Leitmer as the lead for this monitoring project and the great engagement of colleagues, to have comprehensive measure with over 98% data coverage from the indicator, and show the clear trend that EU laboratories across these 3 levels and organization are very capable. Out of a score of maximum 10, the median started around 7 5 years ago and reached almost 8 now. So there's a gradual progress. And importantly, the dispersion of performance overall system index between the least capable countries in the at the bottom here, below 5 years ago, has reached over 6, and from 9 to 10. So there's been a narrowing of the disparities and convergence towards a more comparable capabilities and better connected capabilities across the board and the countries. However, there are also vulnerabilities, and we don't have time to go into the detail, but suffice it to say that what identified as the weakest part of the whole pyramid is its foundation. It's primary diagnostics. And if we look at some targets that compose the diagnostics capacity and capabilities, you can see that the performance score by country is not the same for each component. For instance, regulation, quality control, microbiology services may be strong, dark green in some countries, but the provision of medical policies, testing guidelines, if you like, may be absent in some strongly regulated countries. And the most important, perhaps, which is the testing use, the real life capacity and production of testing and access to testing, also vary quite dramatically between countries. And as we know, can be a very big limiting factor, both in peacetime for endemic disease as well as during a major epidemic. Importantly, there's also been mobilization of advanced IT tool with automated laboratory data from the local information system by machine-to-machine reporting automatically to national surveillance database. And again, Katrin Leimer took the lead here in drilling down that particular capabilities in a survey in 2018, again with all EU member states participating. And what came out is about a third, 9 countries, have put together a system by which, through artificial intelligence and machine-to-machine interface, all the data from the labs, or some labs at least, on diagnostics and stability testing can be gathered on national level and regularly processed to look for cluster or some pan-drug or very multi-drug resistance. So an automatic alert system there. And the CDC is now engaging in a pilot project with several volunteer member states to see how this can be extended to the international level for European alert of antimicrobial resistance and emerging disease. So mastering and starting to put into concrete use the opportunity for digital health from laboratory information. Another thing that's been alluded by Andrea as a contribution that CDC has been working hard, started almost 10 years ago by discussing across the board with experts from Europe and the world. Actually, many of you were participating in this symposium today, and it's wonderful to recap with you on the road we have followed over the last 10 years. And you helped us define what can realistically CDC and the EU disease networks contribute to. And the first thing was try to evaluate whether the whole genome sequencing could do not just for outbreak investigation, but routine surveillance for high priority disease in Europe, and then contributing to defining the standards for enabling data exchange at European level between laboratories for genome-based surveillance and integration with EPI TESI database with the appropriate trainings in the bioinformatics and the cross-sectoral collaboration to build across the various sectors, especially for food and waterborne and zoonotic disease, respiratory and food safety organizations, national and EU level, and importantly, keeping this dialogue going between biologists, scientists, and public health operations. And indeed, it's been very rewarding to see that the gradual acquisition by member states over the past 6 or 7 years of WH-based typing and extensive use to almost all countries now for high priority disease or antimicrobial resistance monitoring has taken place, and this against the odds of in many countries great restraints on the resources available and substantial effort to streamline their workflow and priorities to join these new approaches, which have delivered. I don't have time to come here with more examples, but there are a number of publications from several groups in tuberculosis, in food and waterborne disease, showing that more cross-border outbreaks have been detected through the system, have been more quickly resolved, also for detecting cross-border or regional multi-drug resistant outbreaks as we speak. So this is here to help shorten the delay and enhance the control of this disease. And it's been illustrated too with regards to the good use across Europe of whole genome sequencing capabilities to monitor the dissemination around Europe and the change in different lineages of SARS-CoV-2 during the past 6 months of the pandemic, as illustrated in a very nice Eurosurveillance paper by Eric Alm and about 550, I think, other authors who have contributed to this sequence analysis and shared them in the public domain for real-time data mining and citizen science, if you want, and public health use, analyzing the data as they become available. Of course, to conclude, I'd like to echo some of the points that have been made by Andrea and other guest speakers today, that what have we learned so far about the crash test of COVID-19 and specifically with COVID testing, how much was Europe ready? So first of all, we have to realize that our EU LabCap set of indicators try to capture the element of laboratory preparedness for emerging epidemic disease. And actually, it indicated, I didn't show you, but it indicated quite strong capability for deploying molecular tests for early detection, genome characterization, and tracking by the expert labs, and actually delivered that very quickly and very well. But what was not measured is the plan or system or stockpile or whatever plans and resources put in place for providing the search capacity or the outsourcing beyond this centralized testing that quickly was becoming necessary. So the EU response was to help that with providing the system for EU joint procurement to try and meet the shortage of test material, but it was more a production shortage globally. And of course, launching a number of important R&D projects on rapid diagnostics, which are now delivering very promising tools, as well as ECDC harnessing its existing networks of emerging disease and influenza to task it to COVID-19 diagnostics and characterization, sharing best practices, compiling assay validation and putting them in public domain through publication and open access databases. What's been really quite remarkable, and I think we have to better understand and digest this, is how member state national response to this testing bottleneck has happened and is happening as we speak. And we've seen the most bold political and grassroots initiative to break the bottlenecks and reinvent system or build organization out of the blue for new platforms and players to deliver extended mass testing. The latest one, the Moonshot plan in the UK, in the area of investing £100 billion, that's twice the national defense budget, to provide a testing once a week to every citizen so they can do business as usual in a couple of months. Wow, science fiction. Top down, really very big investment in microbiology, more than I ever dreamt of, I must say. Let's see how this can happen in reality, of course. An example of how reference lab did deliver were ready very quickly was the paper by our lab networks and the CDC colleagues, Chantal Rosken and colleagues, showing that a couple of weeks after the first molecular detection tests were published, and about a month after the sequence was published for SARS-CoV-2, we were able in every country to identify COVID-19 cases, but the rollout to the community and point of care was the issue, as we know. So to conclude, I would like to leave you with some questions to reflect on. Are we, and by we I mean health professionals around this table, and we've been working together, intellectually prepared to change and do what it takes? One element is this fantastic big data, automated collation, AI, integrative analysis, which we need, which is extremely important, but will not suffice. And we should not be hypnotized by technology. I think it will complement, but will not replace field and experimental research and practice to get a good understanding of infectious disease cause and determinants. This very nice concept of evolutionary public health, is this just kind of pipe dream, or can we translate that into real framework to understand how to adapt as host in this fast evolving crash mode of pathogen environment co-evolution, internal to our body and external to the biosphere in this Anthropocene era? Quite a challenge there. I think we have to pay more attention to be on the soft, again, not the war approach, but the cooperation and harmony approach with more restorative intervention strategies to rebalance homeostasis scale from our own gut through diet and prudent use of antimicrobial to the planet microbiomes. We don't really know how they fare, but probably are evolving potentially to a detriment if we don't pay attention. And finally, a bit more kind of our daily practice and the need for outreach between medicine and public health practitioner and to the community. I think we have seen the most fantastic rise of solidarity and crowdsourcing to help bridge the gaps in healthcare, in contact tracing, in testing, and we should build on that. I think we still have to learn, and it's a fantastic period to learn how we can translate the practical wisdom, scientific excellence in this co-creative process. And I'm sure that in the new political will from communities, from policymakers, this is the right time to move into direction and a great opportunity. I'd like to thank you again for sharing my journey through this adventure and wish you the best. Thank you.
Thank you very much, Marc, for such a nice talk, and again showing us the questions that we need to pose ourselves, which are very, very important for the future. I would give the floor to a colleague for a final comment question to Marc, because we're a bit over time. So does anyone wish to make a comment, say something? Maybe one of the presenters of today? Okay. I think you've said it all, Marc.
Inez?
People, yes.
It's,
oh, Mike, yes,
Mike
okay. I
Catchpole,
pass on the floor to Mike Catchpole.
So, Marc, thank you. Mike Catchpole here. I'm, for those that might not know me, I'm Chief Scientist here at ECDC. I just wanted to say what an honor it's been to work with Marc. I've never known anybody so deeply knowledgeable and passionate about their sphere of scientific interest. And I have so many questions I would like to put to Marc, but I give you the choice of two questions to answer, Marc. One is you talked eloquently, as has everybody, about the scientific and technological advances that we've seen over the last few years. But I would be interested to know what you think the professional advances are that we need to see. How do you see these, the development of the professional cadre of public health work changing in the face of this? How do we actually exploit these benefits when we think about how the workforce should change? Or alternatively, although it hasn't really been touched on today, do you see point of care or near patient testing fundamentally changing the face of public health and clinical microbiology in the coming years? I'll give you the choice.
Right. I guess they're somewhat interlinked because the change of technology is also the change of practice, and we have to seize both opportunity and how we best meet the needs of the people at the bedside as well as in communities. But I think clearly what ECDC in its training program can further promote, and I think is promoting and its reflection and other parties like USIC organized by ESCMID in the infection control healthcare based approach is to go towards what Alex called the meta discipline, that is to bring this discipline of epidemiology, of descriptive microbiology, evolutionary biology, and genomics, as well as data science, as well as new approach to modeling of complex ecosystem or dynamic systems, and advanced statistics tools as well that apply both the biology and the epidemiology and the integrated database. So to try to have perhaps less separate category of professional and more people who can navigate across this discipline and master, not just for research, but also for practice, this understanding that all these things are interconnected and you cannot resolve a problem without understanding some of the external determinants, and also make sure that if things change like POCT automation takes away the classical lab, but we would still need to get the microbiology information. We still need to understand what it means, even though we are not going to use pipettes or microscopes to actually find the bugs. So, of course, the training has to follow also the tools that we have. And tools will be more and more decentralized, miniaturized, and of course, owned by the individual and the community. So we have to engage also, not just as professional, patronizing what's good for you, but hearing what they want and how they want to use it. Over.
I don't know, Mark, while you were speaking, did you have the chance to look at the chat? I think you should take a moment to look at the chat because everyone is out there saying thank you and
okay,
paying their respects to you.
My let me see. I don't see much.
Mark, I copied the chat for you and I send it to you later on.
I got the chat, but very small window. Something happened with my screen here.
Yeah. With that, I think we thank all the speakers. We thank you, Mark. We thank everybody that have contributed actively to this session. And we leave the final words of today to Andrea.
Yeah, thank you very much. All the speakers who not only showed their excellence in the field, but also the contact and the inspiration from Mark. I just wanted to say to Herman, if this is the only thing that we two disagree, I'm fine with this. I still think it was a good idea that Mark came to ECDC. And as Ines mentioned, there was a short time where I actually was Mark's manager. And before I met him the first time, I knew his name, but I didn't know the person, Marc Struelens. And what I encountered was someone with a profound knowledge, a vast experience, not only in depth in microbiology, but also in breadth. And I think Mark's talk has shown that he has the macroscopic side of microscopy. And I found him an ally for my idea to bring whole genome sequencing to routine surveillance, very much so. And I think we moved this forward. You helped in the end really to establish ECDC standing in microbiology. To a way that Daniel and all the other colleagues now can build on and take further. So thank you very much for this. I also want to mention that Marc and I have a common passion, and that is hiking in the mountains, especially a bit higher up. When we met in our for our farewell talk here, for obvious reasons, of course, this is now not possible. But when it becomes possible, I wish you that you enjoy a lot of hikes and that you always come back in one piece. So thank you again for all the part of the knowledge that you shared with us. Well, as everybody said, we will miss you. Thank you very much. All the best for your future. And thanks also to everybody who joined us for this symposium.
Thank you so much, Andrea.
Bye.
Thank you so much.
So how do we get out of here?
Truly miss you, Eva here. Bye.
We still have 60 participants, but I think the symposium is over, dears.